The overall goal of this protocol is to use brightfield intravital microscopy and time-lapse video photography to study dynamic neutrophil-endothelial cell interactions during neutrophil recruitment in response to neutrophil chemoattractant in vivo. In this video, we demonstrate the procedures of how to use brightfield intravital video microscopy to visualize and determine neutrophil intraluminal crawling, transendothelial migration, and chemotaxis in cremaster muscle tissue in the mouse. Leukocyte recruitment is the hallmark of inflammation. This recruitment process initiates when leukocytes in the bloodstream start to roll on the endothelium of postcapillary venules at the inflammation site. In response to chemokines or other chemoattractants on the venular surface, these rolling leukocytes arrest on and firmly adhere to the endothelium. After adhesion, many leukocytes crawl on the luminal surface and appear to search for an optimal transmigration site. Following crawling, leukocytes move across the endothelium, a process termed transendothelial migration. Thereafter, neutrophils migrate in the extravascular tissue toward the source of the chemoattractant in the inflammatory site, a process called chemotaxis. This video shows the experiment of how to use brightfield intravital video microscopy, time-lapse photography, and ImageJ to visualize, track, and determine neutrophil intraluminal crawling, transendothelial migration, and migration and chemotaxis in cremaster muscle tissue in response to the source of a neutrophil chemoattractant.
Hello, I'm Lixin Liu from the Department of Pharmacology.